So welcome back. Today we will be doing eukaryotic cells. Now, these bio videos are going to be a little different. I mean, there's only so much I can do, right? Like, it's bio, it's a lot of memorization. So, low-key, these may help me more than you guys because I'm pretty much just going to be talking about these. But if you're down to hear me listen talk about eukaryotic cells, then let's lock in. Okay, so nucleus, right? We need to know the nucleus is 25% of the cell. Okay, fun fact. So again, we all know what the nucleus is, right? But again, surrounded by nuclear membrane. So it's a double membrane that splits up the nuclear environment from the cytoplasm environment, right? And we all know why, like we will need to do that. But we do have something called nuclear pores, which is what allows for transfer of material. So things that are able to go to the nucleus will be filtered by the nuclear membrane, but be allowed to go in like by the nuclear pores. So it's as you can see, the nucleus is sorry about that. As you can see, the nucleus is very selective. So DNA. Obviously, we all know what this is, right? It has coding regions called genes, and make sure you know that DNA is wound around histones. Now, we're going to get to histones very soon, but not yet. So, mitochondria, we all know mitochondria is the powerhouse of the cell. So, mitochondria, obviously, very important. I don't need to tell you that, but a few things, right? So, it has both an outer and an inner membrane. So, the outer membrane serves as a barrier of sorts, okay? And the inner membrane have something that is called cristae, okay? Cristae is the site of the electron transport chain. The electron transport chain happens in the intermembrane space. Again, so the space of the cristae, known as the mitochondrial matrix. Okay. And again, there's a lot more detail that we're going to get into, but just foundation. You need to know that they replicate independently and it can also start apoptosis, which is what we call cell death. Okay. Just a fancy way of saying cell death. So next we have lysosomes. So you can look at lysosomes as sort of like they're like destructors, right? So lysosomes are filled with enzymes that break down different substrates. So they can also cause apoptosis when you release the enzymes, but lysosomes are very important in terms of keeping our body protected, and they're definitely a part of our eukaryotic cells that obviously every part's important, but the enzymes that lysosomes contain really do help break down certain parts of things that we don't need. So next we go to the endopathic reticulum, also called the ER. So there's a few things. So we know that we have two different like ERs per se. It's one ER, but it has two sides, right? So the rough ER, we have ribosomes. And it helps permit translation of proteins destined for secretion to its lumen. So the most important thing you need to know about the rough ER is that it has ribosomes and it helps translate proteins. The smoothie R, what you need to know is that it lacks ribosomes and is used more for lipid synthesis. So the Golgi apparatus you should imagine as, like a post office. So it stores stuff, takes it, modify it. By modifying, I'm talking about post-translational or transcriptional, sorry, modifications, package proteins and lipids, and then it exports them. So Golgi is taking all these stuff, making it into something, and sending it off into the cell. Okay. So next peroxisomes. Now, I think these actually act a little like lysosomes. So they're found in eukaryotic cells, of course. They play a vital role in metabolism and detoxification. Okay, so these contain enzymes that are involved in breaking down fatty acids, detoxifying hydrogen peroxide, and it contributes to a lot of various metabolic pathways. So that's the main difference is peroxisomes are focused more on fatty acids. So last but not least, cytoskeleton. So first off, general overview is it's a dynamic network of protein fibers found within the cell, maintain support, maintain shape, blah blah blah, all that good stuff. So the cytoskeleton is made out of three things called microfilaments, microtubules, and intermediate filaments. You will hear microfilaments also referenced to as actin filaments a lot. So they're thin structures that are in our cell and they're part of the cytoskeleton. They compose protein actin, which plays a vital role in maintaining cell shape, enabling movement, and helping with other cell processes. So these are a pretty big deal. You should know everything about all three of these, but especially microfilaments. So next, let's go to microtubules, which are a more like a circular structure, like it says here, like they're tube-like, hollow, and found in your cytoplasm. We have fluid inside, and it's found in both plant and animal cells. Microtubules support your cell, so they give it a shape, and they also help during cell division. So you'll see that soon as we get to the reproductive system. And last but not least, intermediate filaments. So you can look at these guys as like, I don't know, I've always looked at them as like very structurally sound, right? Like they're strong, exactly right here, mechanical strength and stability. That's really what they do for the cell. They really give the cell like that strength to withstand tension. Okay? So, okay, let me know how that was, guys. Again, obviously this channel is called Joel Does Orgo, so bio's a little out of left field, but definitely comments are needed and appreciated. So chapter 1.3 is going to be prokaryotic cells. So I see you there. Thank you guys. Have a good one.